this is Sean Spencer and Lee Vincent,
Hello.
and we're here to talk about interphase, or meiosis.
Meiosis. Alright. So I'll start off. The first stage of meiosis is interphase G. And interphase G is the period prior to the synthesis of DNA. In this phase, the cells increase in mass in preparation for cell division. Next. In this phase, this is the S phase of interphase. This is the period during which the DNA is synthesized. In most cells, there is a narrow window of time for which the DNA is synthesized. In the prophase 1 of meiosis, the events that occur are the chromosomes condense and they attach to the nuclear envelope. The synapse occurs, which is a pair of hemiglobius chromosomes that line up closely together and triad is formed. Each triad is composed of 4 chromosomes. Crossovering may occur, which is a representation of what we have here currently. Chromosomes thicken and detach from the nuclear envelope. Similar to the mitosis, the centrioles migrate away from each other, and both the nuclear envelope and nucleoli break down. Likewise, the chromosomes begin their migration to the metaphase plate. In this stage of meiosis is metaphase 1. In metaphase 1 of meiosis, the following events occur. Triads align the metaphase plate, and note that the centrioles and the hemiglobius chromosomes are oriented towards the opposite cell poles. In anaphase 1 of meiosis, chromosomes move towards the opposite cell poles. Similar to mitosis, the microtubules such as the nitrofibers interact and pull the chromosomes of the cell poles. Unlike in mitosis, the sister chromatids remain together after the hemiglobius chromosomes move to the opposite poles. In telophase 1 of meiosis, the spindle fibers continue to move of the hemiglobius chromosomes to the poles. Once the movement is complete, each pole has its haploid number of chromosomes. In most cases, cytokinesis occurs at the same time as telophase 1. At the end of telophase 1 and cytokinesis, 2 daughter cells are produced, each with 1/2 of the number of chromosomes of the original parent cell. Depending on the kind of cell, various processes occur in preparation for mitosis too. There is, however, the genetic material does not replicate again. So that would mean that if we had 10 chromosomes before, it would split up and become 5.
In prophase 2 of meiosis, the nuclear membrane and nuclei break up while the spindle network appears. Chromosomes do not replicate any further in this phase of meiosis, and the chromosomes begin migrating to metaphase 2 plate. In metaphase 2 of meiosis, the chromosomes line up at the metaphase 2 plate at the cell's center. The kentochore fibers of the sister chromatids point toward opposite poles. In anaphase 2 of meiosis, sister chromatids separate and begin moving to opposite ends of the cell. Spindle fibers not connected to chromatids lengthen and elongate the cell. Once the paired sister chromatids separate from one another, each is considered a full chromosome. They are referred to as daughter chromosomes. In preparation for the next stage of meiosis, the 2 cell poles also move further apart during the course of anaphase 2. At the end of anaphase 2, each pole contains a complete compilation of chromosomes. In telophase 2 of meiosis, distinct nuclei form at the opposite poles. Cytokinesis occurs. At the end of meiosis 2, 4 daughter cells are produced. Each cell has 1/2 of the number of chromosomes as the original parent cell. This concludes the meiosis stages. We hope that this was informative, and have a nice day.